Assalamualaikum. What is up, guys? This is going to be our second video on the first chapter, cells. And where we left off yesterday was just answering the questions of test yourself. And now we're going to move on to bacterial cell structure. All right. Just like we had plant cell, just like we had animal cell, we have bacterial cell too. And this is part of your course as well. So we need to learn a few details about them. Firstly, bacteria are very small organisms and are not often more than 0.01 millimeter in length. They can only be seen under high magnification of a microscope. Their cell wall is made up of complicated mixture of protein, sugar, and lipids. In specific, they're made up of proteoglycan. Proteoglycan is the specific content which they're made up of. Just like plant cell is made up of cellulose, bacterial cell wall is made up of proteoglycan. All right. Now also you need to also know that inside the cell wall is some of the content of the cytoplasm, and the cytoplasm, as we discussed earlier, has granules, and those granules have lipid droplets, they have energy droplets for food reserves. So it basically gives them energy, right? It gives them energy whenever they require any sort of energy. Now one more thing that is different in a bacterial cell comparatively to an animal and a plant cell is that their ribosomes are comparatively smaller. They have smaller ribosomes comparatively to the animal and plant cell, right? As they have even stated here, ribosomes found in plant and animal cell, but the function of these ribosomes are the same. They perform protein synthesis. If we be really specific, if you further will know that the ribosomes in the bacterial cell are basically 70s ribosomes, and the ribosomes in the animal and plant cell are 80s ribosomes, and hence the ribosomes in the bacterial cell are smaller, right? I hope you understood that well. So that's basically a little more depth knowledge on why the ribosomes are smaller in the bacterial cell. Here's a picture of the bacterial cell over here. We can before we look at the image, first we're going to read a bit more details about them. Now, each bacterial cell contains a single chromosomes, and they're made up of circular DNA. Now, one more difference between a bacterial cell and animal and plant cell is that the bacterial cell has a circular DNA, whereas the animal and plant cells, they have linear DNA. The animal cells have a linear DNA, right? And no, but the plant cells have a circular DNA as well. But for now, what we need to know is that the bacterial cell has a circular DNA, the animal cell has a linear DNA, and the plant cell has a circular DNA as well. Now, whereas the chromosomes are not surrounded by a nuclear membrane, we learned in the previous chapter that there was a nuclear envelope, nuclear membrane around the nucleus. But in the bacterial cell, there is no nucleus, and hence no nuclear membrane either. No nucleus and no membrane of nucleus, right? Whereas there's another smaller circular structure called plasmids, very important. They're also made up of DNA, just like the normal functioning DNA which they have. There's an extra set, you could say, and they're called plasmids. They're a circular DNA. And what is the function, and at least what is the usage of these circular DNAs usually, at least for us? So what scientists do is they use this plasmid and use it for genetic modification. So what is genetic modification? Genetic modification is when you can modify any sort of genetic material and use it for your own benefit or for any purpose. For example, for the rapid production of insulin, right, for diabetic patients, what we do is we take this plasmid out of this bacterial cell, and we can modify this plasmid. For example, this is the plasmid, and we can modify this plasmid by, for example, cutting off this end of the plasmid, right? And then you're left with this much, right? And now you can insert any sort of genetic information you want of your own over here, and then we can insert this plasmid back into the cell. Now what happens here is, one thing you need to know about bacterial cell is that they replicate asexually. Now what is asexual? Now what we all know is that when it comes to human reproduction, or when it comes to plant reproduction, they reproduce sexual reproduction. Sexual reproduction is when two gametes fuse from each parent. For example, one gamete, the gamete from the mother, the gamete from the mother fuses with the gamete from the father gamete and the mother gamete. This is sexual reproduction. Right? When two gametes are coming in and fusing to make a zygote, right? But what we need to know here is asexual reproduction is when there's no two gametes fusing, whereas there's just one gamete which is replicating into another gamete. As we have learned earlier, there's something called mitosis, which is replication of the cell. So it's quite similar as that, but when a cell like bacterial cell is replicating, we would give it asexual reproduction. Is the same as mitotic division. It's asexual reproduction, right? So that's something we need to know, that the bacterial cell can reproduce without having another bacterial cell come and fuse with it. It can just reproduce on its own, right? So when we have inserted this new plasmid with the new genetic information that we need, for example, this codes for insulin, right? Now when this enters back in the bacteria, when the bacteria replicates into thousands and millions of more bacterial cells, hence each bacterial cell which has been produced into a new bacterial cell will have this sort of insulin genetic information. So now this is a way to produce that insulin, right? So that's how scientists use this for the genetic modification purposes, to produce insulin or to produce any other drug sort of thing at a higher level, at a high scale, right? Now to look at a few structures of the bacterial cell. Number one, we have already discussed the ribosomes. They are comparatively smaller than the ribosomes in the animal cells and the plant cells. Number two, we have discussed that the chromosomes have single DNA strand coiled up. Basically it's a circular DNA, it's not a linear DNA, right? And we have an extra part of genetic material called the plasmid. It's also a circular DNA. We have the cytoplasm just as usual as well. We have the cell wall, as we discussed earlier. It's made up of protein, lipids, and other components, but the name that what it basically is made up of is proteoglycan, right? Proteoglycan is what the cell wall is made up of the bacterial cell wall is made up of. We have the glycogen granule reserves for providing energy, and of course as they're replicating, energy is required to replicate, so definitely these glycogen granules will be utilized in this process. One thing that they have not mentioned here but is very important is all of these strand like structures, as you can see here, this strand like structure, these are called the flagella, right? These flagellas are basically proteinous components which help in the movement of these bacteria. These flagella have like a whip-like movement which help in the movement of the bacteria forward or towards any position that they want to go to. These flagella help in that movement, in the locomotion, right? So these are called flagella, right? Now looking at the structures present inside the bacterial cell, as we have discussed, is cytoplasm, jelly-like structures just like previously in animal and plant cells, surrounded by cell membrane. It contains structures like ribosome, circular DNA, and plasmid, cell membrane partially permeable. just like the previous cell membrane, which was also partially permeable, and same function as previous cell membrane of the animal cell and plant cell, prevents cell from escaping or prevents cell from entering the cell as well. Circular DNA, circular chromosome inside cytoplasm controls cell division, controls cell development. Same functions of that of as of a nucleus inside an animal cell. Same functions. Controls the cell division, controls cell development. What does that mean? What the cell will become in the future, what the cell will mature into in the future, cell activities, whatever is going on, chemical metabolic activities inside the cell. Plasmid is an extra genetic material present in the bacterial cell. It contains genes, carry genetic information to help the process of the and reproduction of the bacterium. It also helps in the reproduction of the bacterium, survival and reproduction. Ribosomes are small circular structures, helps in protein synthesis. We have discussed what the ribosomes do earlier. Cell wall, a tough non-living layer as usual, not made up of cellulose, very important. They may ask you in the exam, is the cell wall of a bacterial cell made up of cellulose? That's not true. It's made up of what? Proteoglycan. And that proteoglycan is basically structures which is a combination of protein, sugars, and lipids. Protein, sugars, and lipids. Good. So basically, the cell wall does the same function. It prevents the cell from bursting, number one function, and allows water and salts to pass through freely permeable. Easy peasy. Now, something I need to mention here is the practical work. I'll be creating a different playlist to teach this practical work. So I'll be creating another one playlist. So you could refer to that. But as of now, we're going to be just learning theory-based knowledge. And all these practical cases we're going to be learning in that playlist, whether it be as of now here, whether it be looking at a cell under a microscope, or whether it be magnification of a cell as well. These all will be done later on. All right, so let's test ourselves here. Just a quick revision. How is a bacterial cell different from a plant cell? As we discussed earlier, number one, the cell wall is made up of proteoglycan. That's how, number one, it's different from the plant cell. Number two, it has an extra DNA material which is called plasmid. Number two, it does not have a nucleus, no nucleus, and hence there's no nuclear membrane. Very easy. Bacteria and plant cells both have cell wall. In what way are the cell walls different? As we have discussed, bacterial plant cell are made up of cell wall are made up of proteoglycans, whereas the plant cell are made up of cellulose. Make large drawing, one labeled. This is easy, and this all is about magnification, which we are going to learn later on in the other playlist. Now moving on to specialization of the cell. All right. Now through the past chapter and even here, we have been learning that the nucleus controls controls cell development. Now this cell development is related to the cell specialization, how the cell is going to develop into the specialized cell. Now you need to realize every cell has its own different type of function, which it is appropriate for its own type of environment, right? And that's why it's called a specialized cell. Here's written, when the cell is finished dividing and growing, most become specialized, which is basically specialized to their function. They do one specific job. They develop distinct shape according to their site, according to their function. Special kinds of chemical changes take place in their cytoplasm, right? Easy peasy. We have an example here. As they said, the changes of the shape of the chemical reaction enable them to carry out their special functions, as I described earlier. They give an example. Red blood cells and root hair cells are just two examples of specialized cells. The specialized cells carry out special functions in organisms called division of labor. Now, a very important term that they've used here is division of labor. Now, what does that even mean? What that means is that all of the different types of function that different types of cell would be doing inside an organism has been divided ये वाला, for example, the red blood cell carries oxygen. For example, the blood carries all the nutrients, and so on and so forth. There's all different types of cells have their own function. The root hair cells helps in absorption. We're going to learn further about all the different types of cells present, but the point is that all the work has been divided into different types of categories where all the different types of cells are performing their own function, whereas it's not like that one cell is performing all of these functions. That's not true. It's every cell has its own specific function. For example, this cell has its function, so that's how it is, right? So that's called division of labor. And similarly in right now we were talking about different cells, but we have also learned that cells, inside the cells in the cytoplasm, they have organelles have their different function too. Now that's also a division of labor, but what we will the proper term we'll use here is it's called division of labor within a cell. So inside a cell, how the functions are divided, like mitochondria to produce energy, ribosomes for protein synthesis, and so on and so forth, nucleus for cell division. So even inside the cell there's division of labor, and we could say division of labor within the cell. So these are some really great interesting terms that are present within our body. Now the first example that they have given here is ciliated cells. Ciliated cells are basically hair like, these are basically cells, right? The usual cells as that we have, but at their top surface, they are their apical surface. Apical is basically their top surface. There's cilia, hair like projections present, as you can see, right? And the example that they have given here is that in your windpipe, from your nose and your windpipe, where your air enters, there are tiny cytoplasmic hair-like extensions called cilia, continuously flicking movement, right? Which helps in secreting the mucus, carries dust and bacterial through the bronchi. And for example, let's suppose this is your windpipe, right? And this is your mouth. All right, it's a very bad mouth I've made here, but for example, this is your windpipe. And for example, here are the cilia present, these hair-like projections, and there has been some dust that has entered into your mouth, and it has entered into your windpipe, right? So what this cilia does is it does its for example, this is the cilia, right? So it does its flicking type motion, which helps it push the dust material up the respiratory tract, and push it up the respiratory tract and then outside of the mouth, or outside of the nose as well, right? So that's what it does. It does a flicking movement which helps trap the mucus and the dust particles and help it to secrete it out of the mouth or out of the nose, or even it might push it into the esophagus and hence goes into your stomach and is digested there. So that's what it does. It basically prevents all the bacterial cell, all the dust to enter into your lungs, basically, right? Here, away from your lungs. It's trying to keep all the bad material, dust, bacterial cell, and so on and so forth away from your lungs, right? It also secretes mucus here, by the way. Secretes mucus. These cells, you see these cells over here? These secrete mucus. So these mucus basically trap the dust particles and bacteria. Now once they, for example, this is the bacteria or the dust particle, this has been trapped. Now what the cilia does is it does its flicking motion, right? It does its flicking motion and hence pushes this out of the windpipe and into the mouth and then outside the mouth. Now second example we have here is root hair cells. This is present usually in a plant cell. Their function is to absorb water and minerals from the soil. They have hair-like projections. Each cell penetrates between soil particles and offers large absorption surface. Now, one thing you need to realize here is, just like they described earlier, every cell has its shape and size and is according to its function. Now, the reason why the root hair cell has such a hair-like extension is so it increases the rate of absorb. Increase is the rate of absorption. Absorption. Rate of absorption of what? H2O, salts, and so on and so forth, so that they can enter into the root hair cell at a faster speed. And why is that? Why does it have to be a hair-like extension, and why does it have to be like this, which helps it absorb faster? Well, it's because when you increase the surface area to volume ratio, then it increases the rate of absorption. Hence, as you can see, the volume here is very low, but the surface area which is intact with the outer surface is very, very high. Hence, it helps in further absorption and faster absorption of all these materials. And we have already learned that the cell membrane is able to control which dissolved entered the cell and leaves the cell very easy. Palisade mesophyll, we have been introduced to this in the previous discussion as well. They're found underneath the epidermis of the plant leaves. Epidermis is basically the top layer, the epidermis, right, of the plant leaves. And they're columnar, quite long. They have chloroplasts to trap sunlight. We have discussed that earlier. Their function is to make food for the plant by photosynthesis and using carbon dioxide and so on and so forth. We already know photosynthesis. It traps the sunlight, uses carbon dioxide and water, produces oxygen and energy. It's very, very easy. Now moving on to nerve cells. Now these nerve cells are basically inside your human body, inside the animal body, right? And what these nerve cells do, these are specialized cells, as we have discussed. They help in conducting the impulses fiber from the brain and the spinal cord to the periphery. For example, let's suppose, I'll give you a very easy example so you could understand this whole scenario. Let's suppose this is your foot, right? And basically someone places a hot object onto your foot over here, right? A very hot object onto your foot. You will get a sensation over here that there's a hot object and I need to remove my foot from the place so that it stops burning my foot. Now what happens here is these nerves are going to sense that from here. These nerves are going to sense that from here, and they're going to send that message up into the brain, and then the brain is going to give a message to remove the foot. Now this message sending and message receiving is done by the help of nerves. Nerves are basically the road which this message travels on. Just like a car travels on a road, just like that, the message to remove the foot travels inside the nerves. So it travels in the form of electrical impulses from one neuron to the next and into the brain, and then so on and so forth from the brain to the periphery. And the fibers are often very long. They connect distant parts of body to the CNS, as I said. Example, the foot till the CNS, which is the brain and the spinal cord. For example, here they give the foot and the spinal cord. Okay, a few components we need to know about the nerve cell is, as usual, it has a nucleus as it is a cell. Not all cells have nucleus, keeping that in mind, but this definitely has a nucleus, has cell body having its own organelles and contents present within it. And something very new here is it has a nerve fiber, a very, 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 very long nerve fiber. In specific, this nerve fiber is called an axon, right? This nerve fiber is called an axon. And the nerve impulse, for example, these hair-like extensions are called the dendrites. These dendrites detect the stimulus. For example, the hot object was placed, right? Now this detects that stimulus and sends it up into the cell body, and then hence sends it up the nerve fiber, right, which is basically the axon. So this electrical impulse, this electrical message travels up the axon into here, and this is basically called the axon terminals. The axon terminals, which transmit the information to the next part, which could be maybe another nerve cell exactly same like this. There could be another nerve cell present over here, for example, another nerve cell present over here detecting this message to the next, or this could go to the brain, or from as we mentioned earlier, from the brain, as you mentioned earlier, from the brain a message could come back to the foot to move the foot. So this message that's coming back to the foot, for example, the axon terminals could be here. These axon terminals could be here and could act onto the foot muscles and make the foot move away. So that's how it basically works, right? Now moving on, very important thing, you all will learn this throughout your A level, O level. RBCs. RBCs are basically present within your blood, right? And they have no nucleus. One very special thing about these RBCs is that they don't have any nucleus when they are mature, whereas when they're like they're developing and during the process of maturation, they do have a nucleus, but by the end of it, they lose their nucleus. And we all know that RBCs have a lifespan of 120 days. And why is that? Why is there a lifespan for these cells? Whereas we discussed earlier that nucleus inside a cell helps us to replicate, and once that nucleus is not present, it can't replicate. Hence, it has a lifespan of 120 days. That basically gives us a very good reason of why it has lifespan, and it cannot divide. It loses its nucleus. That's why. The tiny disc-like cells contains red pigment called hemoglobin. This readily combines with oxygen. Function of now inside this RBC, which gives it basically the red color, is the hemoglobin present. This hemoglobin pigments. These are also pigments, just like chloroplast had green pigments called chlorophyll. RBCs have red pigments called hemoglobin. What this hemoglobin does is that it basically binds with O2. It binds with oxygen, and what it does is within your blood, for example, this is the blood capillary, and here are your RBCs present, and inside that is your hemoglobin present, right? And inside that is your hemoglobin present, and that's carrying your O2, and hence the O2 is being transported inside your blood vessels to the rest of your body. And we all know oxygen is required by all cells to provide energy for the cells to reproduce, and so on and so forth. So this hemoglobin helps in the binding of oxygen and helps it transport through our body via the blood. Okay, so hemoglobin is a red pigment, helps oxygen bind to it and helps in the transportation of the oxygen. All right, and we have to also know that RBCs do not have a nucleus, and that's exactly why they have a lifespan of 120 days. Now moving on to sperm cell. Now these sperm cells are produced within a male body, a male human being. It's a male sex cell and is basically released upon ejaculation of the man. A few structures that you need to keep in mind is, you can see that there's a nucleus, and nucleus of course is going to carry the genetic information, and there's an acrosome here, all right, at the tip of this sperm cell, and at the middle part of the sperm cell this is called the midpiece, and we're going to talk about exactly what that midpiece does, and we have a tail here as well. All right, perfect. Now let's describe what these things are. So in front there's a cell. The cell is oval shaped and it contains a nucleus, of course, and the nucleus carries genetic information. We have discussed what the nucleus does. There's a tip called acrosome. Now what this acrosome does is, the acrosome upon, for example, this is the egg. This is the egg of the female, right, waiting to be penetrated by the sperm. Now when the sperm is about to penetrate the egg of the female, it releases a few enzymes or digestive enzymes, and by the help of acrosome, and hence it breaks the wall of the or the more appropriate word would be, hence digests the wall of the egg of the female, and it then enters into the egg and then fertilizes with it, right? That's what the acrosome does. It digests the cell, it secretes enzyme and digests the cell around the egg and the egg membrane. The midpiece is packed with mitochondria for energy, for movement. Now here, as you can see, this mitochondria is basically is the midpiece, right? Mitochondria is basically the midpiece. Now you need to know, I'm sure you guys have seen animations of the sperm cells moving around in the womb, like some animated films or whatever. Now how does this sperm cell move around? Like how does it move around and stuff? That's done by the help of the energy that has been provided by the mitochondria present in the mid piece of the sperm cell. That's how the cell moves around in the womb of the mother or inside the male human body as well. And also the tail, which is at the last part of this, helps in the movement of this who helps in the movement of the sperm cell as well. It has also a whip like action, just like the cilia had a whip like action, helped in the movement of the dust particles up the respiratory tract. Just like that, the tail has a whip like action. You could see like a whip like action which helps in the movement of this sperm cell forward, right? So there's two things that help the sperm cell move. Number one is the energy that's been provided by the mitochondria, which is present as in the mid piece. Number two, the tail, which has a whip like action movement, right? And once which helps enabling the sperm to swim. Their function is reproduction achieved by fertilization of the egg. As we discussed earlier over here, that the bacterial cell performs asexual reproduction, whereas the animal and plant cells perform sexual reproduction. And I discussed for sexual reproduction you need a mother gamete and you need a father's gamete. The father's gamete is the sperm cell, right, which is going to travel into the mother, whereas the mother has the egg cell. So the gamete which I'm talking about in the father cell is basically this nucleus present over here. This is the gamete. Gamete is basically the genetic information that has been carried by one of the parents, which helps in reproduction. And once the father and mother gamete fertilize, meaning they fuse with another, they form the new generation, which is called the zygote. Perfect, easy, nothing is difficult, right? And here lastly is the egg cell that we have been talking about, which is which is far larger than the sperm cell and are spherical, right? They have a large amount of cytoplasm. Here's the cytoplasm, as you can see, containing yolk droplets made up of proteins and fats. Important content. The nucleus carries genetic information. Their function of the cell is reproduction. Here is the nucleus carrying the genetic information. Once the let me draw this. My drawing is not that good, but let's suppose that this is the sperm cell about to, and this is the tail. This is the mid piece, and this part is the nucleus. This dotted part is the nucleus, right? Once this acrosome on the tip secretes its digestive enzymes, this part will disappear and will be digested, and hence the nucleus is going to fuse in with the nucleus of the egg present inside the female. So that's basically how the fertilization will occur, and that's how a new generation will be created, a new baby will be formed, right? Very easy peasy, nothing is difficult here. It's all very interesting as well. Now test yourself, right? They ask, in what ways does the red blood cell shown in figure 1.16 differ from most other animal cells? Very easy. We already have identified number one, the nucleus. It has no nucleus. Number two, it has hemoglobin. It has red pigment called hemoglobin, which helps in carrying oxygen. These are the two ways that it's different from usual animal cells. Usual animal cells have nucleus and usual animal cells do not have hemoglobin. It's very easy. Now one more thing you need to know about red blood cells is their shape. Their shape is a biconcave shape. Now what this shape does is it helps in the movement of the red blood cells into the small blood vessels and small capillaries. For example, let's suppose there's this blood vessels, and there's this very tiny capillary which the red blood cell has to pass through, right? Now if the red blood cells did not have this biconcave shape and it was not flexible, it's flexible, it helps it become flexible. If it was not flexible, it would not be able to pass through this blood vessel, whereas hence because of its shape and its flexibility, it's able to pass through very tiny capillaries and very tiny blood vessels as well. So this is very important as well. Now here's another question: why does the cell shown in figure 1.7 on page 4 appear to have no nucleus? Oh, they're saying why the trans section does not have a nucleus. This one, it's very easy, because if I take a cross section from here, the transverse section, I'm only gonna get this view, and hence I've missed out on the nucleus which is present. It's not that the nucleus is not present. It's actually the way I'm looking at the cell. We already described, if I was looking at it in a longitudinal section, then I definitely would be getting a view of this nucleus present here. Whereas the reason why in this image I'm not getting a nucleus present here is simply because I have taken a horizontal section, and when I take a horizontal section, this is all I see. The top layer is all I see, and hence beneath the top layer is the nucleus, and we cannot see that. That's why here, if we had gone a bit more further down, there would be the nucleus present, but that's not the case here. That's why we cannot see the nucleus. It's a very simple question. I really thought they were asking about this topic, but they really made us go back up. Okay, so the next chapter will be tissue, organ, and organisms. I'll be discussing this in the next video. Until then, catch you next time. Hope you guys enjoyed this video. Hope you guys found it fun, interesting, and I hope you guys understood all of these concepts. I know it's all new for you. All of these things are new for you. It's all right. Throughout the chapters, I will be keep on explaining, keep on revising all of these things so that they sit into your brains and that you absorb it really well. All right, this is it for this time. Thank you.